Week 4 mini exam video, prokaryotic DNA replication. Replication that occurs in prokaryotes starts at the origin of replication. The origin of replication is where the DNA begins to open. An initiator protein will bind to this origin of replication, which causes DNA to unwind. The unwound portion, shown in the middle of the DNA, is single-stranded. To facilitate the unwinding of the DNA, helicase binds to the DNA and breaks hydrogen bonds along the DNA in order to separate the DNA strands, creating a replication fork. Once the strand is separated into single strands by the helicase, single-stranded binding proteins bind to the strands to serve as protection as well as to ensure that the strands stay separated from one another. The topoisomerase, in this case gyrase, then binds to the DNA ahead of the replication forks. The topoisomerase is an enzyme whose purpose is to relieve the DNA of supercoiling. A DNA double helix is antiparallel. This means that one strand is 5' to 3', while the other strand is 3' to 5'. DNA is synthesized 5' to 3', and the template DNA is synthesized the opposite of this, which is 3' to 5'. The strand that is synthesized towards the origin of replication is the leading strand, and the strand that is synthesized away from the origin of replication is the lagging strand. Before DNA polymerase can create new DNA on the single strands, RNA primase must lay down the primers on the single strands for nucleotides to attach to the 3' OH end of the primer. After the RNA primers are in place, the DNA polymerase III will then add nucleotides to the primer. DNA polymerase I then serves to replace the RNA primers with DNA. If the DNA polymerase I fails to function properly, then RNA primers will still be present on the DNA even after it is synthesized. The lagging strand goes indirectionally away from the replication fork and within each fragment requires a new RNA primer to start the synthesis. On the other hand, the leading strand just needs one primer. As we can see, the primers have all been replaced by DNA. The lagging strand on the bottom produces fragments that are individually synthesized, known as Okazaki fragments. The fragments are then sealed by ligase to create one continuous strand of DNA.